Hello and welcome everyone. In this podcast, we're going to begin our discussion of transport across a membrane. So let's begin by drawing a cell to introduce the problem here. Up to this point, we've talked about membranes, as I'm drawing here this plasma membrane. We've talked about reactions in a cell caused by enzymes. We talked about various organelles in the cytosol here, the nucleus, the mitochondria, and we'll come back to these later on, but we did mention them, and some others. We also talked about, within these membranes, specific proteins that could be on the outside, could be on the inside. They might go through the membrane. We talked about the membrane itself, how within the membrane we had a hydrophobic portion, and on the outside of the membrane we had a hydrophilic portion. We talked about how these lipids here could have sugars attached to them or a phosphate group. But we haven't really addressed how this cell is going to survive. Cells don't exist in a vacuum. They exist within an environment. They have to have some way to exchange molecules with the environment. Potassium, sodium, calcium, oxygen, CO2, glucose. These all are outside the cell, and they're also all inside the cell. So there has to be a mechanism to send these ions back and forth to maintain a proper homeostasis within the cell. Some of these will be easier to move across the membrane. Some of them will require more energy, more work. But it's important that the cell has a way to exchange with the environment these particular ions and other ions. So that goes hand in hand with this second one, and that's regulating the concentration of ions in the cytosol and organelles. So proper concentration of these ions has to be maintained on the outside of the cell, in the cytosol, and within these various organelles. The next one here is import nutrients. And I already wrote that here with glucose, but it could also be amino acids that have to move into the cell. So there has to be a mechanism to bring in nutrients into the cell. Likewise, we have to export waste from the cell. These various enzymatic reactions generate some sort of waste product, and those waste products have to be removed from the cell in order for the cell to survive. So over here I mentioned at the very beginning is that we have to have a mechanism to exchange ions across the membrane. Sodium, potassium, calcium, oxygen, carbon dioxide, and other ions. So let's consider the type of solute in order to understand whether or not it will easily cross the membrane or if it will require an investment of energy to do so. So let's list some of these factors that affect movement across a membrane. Okay, the first one is size. That might not seem too surprising, but we know that smaller molecules diffuse easier than larger molecules. The second factor is polarity. Nonpolar molecules diffuse easier compared to polar molecules. And the last one is whether or not the molecule is ionic. Does it have a charge? And that's because the membrane is very impermeable to ions. So let's over here draw a membrane, and let's start with small hydrophobic molecules. This would be like oxygen, CO2, If we look at our rules over here, it's small and it's hydrophobic, nonpolar. So these will easily diffuse across the membrane. Next, let's consider small, uncharged polar molecules. This would be like water and ethanol. These will also diffuse across the membrane. Next, let's look at larger, uncharged polar molecules. This would be like glucose, nucleotides, and amino acids. These are not going to easily move across the membrane. They can still get across the membrane, and we'll talk about that in a moment, but they're going to require some extra help getting across the membrane. And lastly, let's consider our ions. These could be sodium, potassium, calcium, chloride, and lots of others. And even though these are very small molecules, this membrane is impermeable to ions. So they will not be able to diffuse across the membrane. Not easily get across. They will be able to get across, but they will require special proteins to help them get across, which we'll talk about in just a moment. Now I want to talk a little bit more about these ions and consider some of the relative concentrations inside the cell and outside of the cell. Okay, so let's talk about ion concentration. Your book gives specific concentrations of these ions. I want you to know the relative concentration, meaning are particular ions in a higher concentration within the cell or outside of the cell. And so let's make a little table. Here we will have our ions. Here we will have inside the cell. And here we will have outside. And let's talk about these ions: sodium, potassium, magnesium, calcium, and then chloride. Sodium has a greater concentration outside the cell, so I'm going to just give it a plus with the concentration symbol around it. And sodium inside the cell is lower concentration in comparison to outside the cell. Potassium is higher in the cell compared to outside of the cell where it is lower. Magnesium, it's a very low concentration both inside and outside, but it doesn't take a lot to do what it needs to do. Outside of the cell, it has a higher concentration. It's about 4 or 5 times higher. Inside the cell, then it would be lower. Calcium is higher outside of the cell than inside the cell. Now there are parts inside the cell, like the ER, that's going to have a very high concentration, but we're just talking about in general inside of the cell. And then the last one is chloride. Chloride has a higher concentration outside of the cell compared to a lower concentration inside the cell. So don't worry about concentrations specifically, but you should know the relative concentration, higher or lower, inside or outside of the cell. What you'll notice is that potassium is the only one of these that is in a higher concentration inside the cell compared to out. Everything else has a higher concentration outside. Maintaining these concentrations sometimes will require the cell to expend a little energy, or maybe even a lot of energy, in order to maintain this relative concentration of these ions. Now what I want to mention now are some of the proteins that are involved in membrane transport. Getting some of those molecules across the membrane that are going to have a harder time moving, like the ions or the larger molecules, may require some help from these proteins. So let's draw our membrane. And I'm not going to draw the fatty acid tails here, but you should know that they are there. The first type of protein I'm going to talk about that's involved in transportation is called a transporter. I'm going to draw it like this. Now I want to draw this molecule that is going to move through this transporter to get from this side of the cell membrane to this side of the cell membrane. So here's this solute. It binds here into an active site, or a protein binding site. It causes a conformational change of this transport protein, causing this end to open up now. And now that solute that was on this side here will now move towards this side because of that conformational change of these proteins that forces it across the other side. In doing so, you can see how this solute never had to touch this membrane. It never had to find a way to dissolve in this nonpolar environment. It was able to bypass the membrane by using these transporter proteins. And I want to write that word I said here because it's an important word when we're considering this kind of movement. Conformational change. It requires this conformational change once it binds the solute. I also want to say that transporters are fairly specific about what they're moving across the cell. Hello, just a quick editing note. When I say specificity there, I'm talking about the same kind of specificity that an enzyme has. So only very specific solutes will bind to specific transporter proteins or carrier proteins. They won't just grab any protein to allow them to get transported. This is the way we ourselves will regulate what gets in and out of a cell by having this specificity of solute to transport protein. All right, let's go back to the video. Next, I want to talk about channel proteins. I'm going to extend the membrane a little bit, and let's go ahead and draw this channel protein. Using channel proteins, certain solutes can now freely pass between this channel. Now that we understand the proteins that can be used to move solids across the membrane, I want to now talk about the different ways or the different mechanisms that cells will use to move solids. Let's call this board here mechanisms of transport. So let's draw our famous membrane here. So I want to make sure we talk about simple diffusion, passive transport, and active transport. We'll start with simple diffusion. When we talk about simple diffusion, we mean that molecules can move across the membrane without help, meaning proteins don't have to help move it. We say that this kind of movement moves down the concentration gradient. And what we mean by that is we're going from a low concentration here to a high concentration here. And an example of this might be oxygen. Oxygen would move across the membrane using simple diffusion. So over here you may have several oxygen molecules, a high concentration of oxygen, and a relatively low concentration on this side of the membrane. So it's going to go from a high concentration to a low concentration. So it's going to diffuse across the membrane. Remember, polar molecules can't do this because of this hydrophobic area of the membrane. Okay, passive transport. At one time you may have learned this as facilitated diffusion, but this is a situation where molecules need proteins for transportation. Like simple diffusion, passive transport moves down concentration gradient. Now, passive transport can either use our channels, like so, or it could use one of these transport proteins. For this, we'll use an example solute that we'll just call I. So let's say solute I is in a high concentration on this side of the membrane and a low concentration over here. Solute I will bind into this transporter. Conformational will change occur, allowing it to be released over here until you end up with roughly equal concentrations on both sides of the membrane. Glucose is a good example for one of these channels, and it will move through the channel from a high concentration to a low concentration. Okay, now let's talk about active transport. This is a situation where molecules need proteins. So in this sense, they're like passive transport. So they need protein and energy. Passive transport and simple diffusion do not require energy. So this is the first one that requires energy. So it needs this energy for transport. We say that this moves against the gradient. You could also say it moves up the gradient, but we usually say it moves against the gradient. It will also require a transporter. Channels won't work so well with active transport, so they require these transport proteins. And so let me draw a transport protein. And good example of this would be the ions. So maybe chloride we'll talk about. And it's going to go against the gradient. So in this example here, we're going to have a low concentration of chloride on this side of the membrane and a high concentration on this side. If it was going to be by passive transport or by simple diffusion, you would expect these chlorides to go this way. But this kind of transportation goes against the gradient. So chloride's going to bind from the low concentration. It'll bind to this transporter, change the conformation, and release it on this side, making the concentration of chloride, in this case, even higher on this side. I want to label these here real quick. So I'm going to erase the bottom part here. And I want to talk a little bit more about passive transport. And I'm going to talk about some of the factors that influence how it moves. One of them I've already talked about, and that is the concentration gradient. And when we think about the concentration gradient, the only thing we're thinking about is the concentration of the solute on one side of the membrane compared to the other side of the membrane. And it's always going to go from a high concentration to a lower concentration. The other factor that drives passive transport is the electrochemical gradient. So what I'm saying with the electrochemical gradient is the amount of charged particles on one side of the gradient compared with the amount of charged molecules or charged solutes on the other side of the gradient. So we would expect them to move from a large number of positive charged solutes to a smaller number of positive charged solutes. Now I want to talk a little bit more about active transport. As I said here, it's going to move against the gradient, the concentration gradient. So it will move from a low concentration to a high concentration. These would be endergonic reactions. So just to review what we mean by that, what we're saying is that the delta G is greater than 0. So this will not be a spontaneous reaction. It will require energy. So this will require the use of ATP. So it's needed for active transport. Okay, on the last slide, we talked about the importance of ATP in moving solutes across a membrane where it's going from a lower concentration to a higher concentration. So let's talk more specifically about a very important ATPase that's involved in membrane transport. And this is the sodium, potassium, pump. So let's begin by drawing our sodium-potassium pump inside of a plasma membrane. Here's our membrane in orange. And then let's draw the sodium-potassium pump, something like this. And let's remind ourselves that on the outside of the cell, there are lots of sodium ions. And on the inside of the cell, there are fewer. So this is inside, or we might call it the cytoplasm. And up here is outside the cell. And we might call that the extracellular space. And I should also indicate that potassium is higher on the inside of the cell compared to the outside of the cell. But for this pump to work, we need to push sodium to the outside of the cell, even though there's more of it out there, so it's going against its gradient. And we need to have potassium come inside of the cell. So how are we going to make this endergonic reaction move forward? Now, even though there's fewer sodium ions on the inside of this cell, there's even fewer residing inside of this carrier protein. So these sodium ions will move into this area here, 3 of them to be precise, will bind to these carrier proteins. In doing so, these sodium ions activate an ATPase that's part of the sodium-potassium channel. When this becomes active, ATP, I'm going to erase these potassium ions. We'll put them down here just so we don't forget that there's a lot of them. But I need a little more room here. So this pump now is activated by the sodium, and it converts ATP to ADP. That extra phosphate is transferred to the sodium-potassium pump here. This causes a conformational change of this pump. And what happens is this channel will now open up in such a way that the bottom part now closes off, and these potassium ions that were right here can now be pushed into the extracellular space, even though there's greater concentrations here. At the same time, now that this channel has opened up, potassium can now diffuse into these carrier proteins of the potassium-sodium pump. Then what happens is this phosphate is removed. And when that phosphate is removed, there is another conformational change that turns the sodium-potassium pump back to its original state, thereby releasing the potassium that were once on the outside, but now because of the conformational change that has occurred because of losing the phosphate, now the potassium can move into the cell, even though there's a high concentration of potassium here. Now this pump is ready to receive more sodium ions and start the whole process over again. And this is all possible because of our ATPase activity that's found within the sodium-potassium pump. So why is this important? Well, let's talk about a few reasons here. First of all, this pump is found in most animal plasma membranes. Proper potassium and sodium concentrations are needed for many cellular reactions. Sodium and potassium are needed for something we haven't talked about yet, but we're going to talk about it next. And it's something we call secondary active transport, which is also called co-transport. And we'll come back to that in just a moment. And the last thing that I want to tell you is that, and this is just evidence of how important it is that about 30% of all ATP made used in this pump. That's a lot of ATP that our body makes, that our cells make, that is used just for moving potassium and sodium back and forth. Okay, now let's finish things up here by talking about secondary active transport. Okay, so let's go ahead and talk about this secondary active transport, which is often just called co-transport. I think the easiest way to talk about and to understand secondary active transport is to think about digestion. And so I'm going to draw an epithelial cell in our small intestines. One of the goals is to get glucose, which I'll just abbreviate GLU, from the lumen of our small intestines through these epithelial cells and out the other end to enter our circulatory system so that glucose can be moved to other parts of our body. One of the problems, though, is that outside of this cell, glucose is low. Glucose in the cell, relatively speaking, is high concentrations. And again, on this side of the cell, glucose is also low. So to go from the lumen of our intestines to inside the cell, it's going to have to go against its concentration gradient, and that's going to require active transport. Now in this case, it's not going to directly use ATP. It's going to use co-transport. Now let's bring back our good friend sodium. Remember what I said on the last slide was that sodium on the outside of the cell was in high concentration. It's also in high concentration on this side of the cell as well. And remember, sodium inside the cell is low. So we talked about the sodium-potassium pump, but I'm going to describe a different transport protein now. And I'm going to flatten out this membrane here just for ease of drawing. There is a pump on this side of the plasma membrane. Now remember, unlike the last whiteboard, sodium is going to want to move in this direction from high to low concentrations. And so sodium is going to bind to this glucose pump. In fact, let's go ahead and give it that name here. Because sodium binds here because it's trying to get across this membrane. And remember, being an ion, it can't freely diffuse across it. It needs a protein to move it across. So it binds to this glucose pump. When it binds to this glucose pump, there is another conformational change to the protein. So again, sodium binds to this pump, causes a conformational change. This allows glucose now to bind to this pump. Before it couldn't, but now it can because of that conformational change. When that happens, both sodium moves into the cell plus glucose. Glucose is going from a low concentration to a high concentration. And it's able to do that due to co-transport. Sodium wants to come in the cell because of it going from a high concentration to a low concentration. And it brings along with it glucose. Now glucose, once in the cell, the concentration is even higher now. So there's going to be another protein here, another transporter, that glucose will bind to. And again, that conformational change occurs, allowing glucose to leave the cell from a high concentration to a low concentration. So this would be an example of facilitated diffusion. And this is all because of co-transport. Sodium, using facilitated diffusion, moves into the cell, brings with it glucose. Glucose then can enter the cell via active transport. And now it leaves the cell into our blood system using facilitated diffusion. Now, you might imagine one problem here. We're pumping in all of the sodium. At some point, the sodium is going to get too high here, and now no more sodium can come in, and therefore no more glucose. That's where our good friend, the sodium-potassium pump, comes into play. And that's going to take sodium and move it against its concentration gradient from a low concentration to a high concentration. And then, of course, move potassium in this direction as well. So the movement of glucose from the lumen of our small intestines through the cell and into our blood system is an excellent example demonstrating several things we learned about. We have an example of facilitated diffusion, conformational changes, a new example of co-transport, active transport using potassium and sodium pumps, and then again, facilitated diffusion moving glucose out. Okay, let's have a quick summary here. of what we talked about on the past 2 podcasts. The first podcast, we talked about membrane proteins. We talked about transmembrane proteins, peripheral membrane proteins, and lipid-linked proteins. We talked about how experimentally we could remove proteins from the membrane. We talked about the effects of detergents, salts, and enzymes. We also compared the fluid mosaic model with the sandwich model, and how that the use of freeze fracture electron microscopy, we were able to determine that the most likely model for the way the membranes are formed is by the fluid mosaic model. Then in this podcast, we focused on transport. We talked about simple diffusion, or sometimes we call that passive diffusion, facilitated diffusion, and active transport. We compared and contrasted carrier, or sometimes we call them transport proteins, versus channel proteins. We talked about active transport requiring ATP, and we focused heavily on the sodium-potassium pump. And then we ended with co-transport as an example to think about how to get glucose through our intestinal cells and into our bloodstream. All right, that sums up most of what we talked about. Of course, there were a few other things. But if you have any questions at all, please come see me. If not, I'll see you in class. Bye.